Hi, I'm Miracle. Here is a comprehensive guide on how to run both polyacrylamide and agarose gel electrophoresis. Both techniques separate molecules based on size, charge, and shape. Let's talk about agarose gel electrophoresis. It is used for separating large nucleic acids, that is DNA and RNA. Materials needed for this experiment are agarose powder, buffet, DNA samples, loading die, gel casting tray and comb, electrophoresis chamber and power supply, UV trans illuminator. Number one step is to prepare the gel. Weight out the appropriate amount of agarose and add it to a flask with the electrophoresis buffet. Heat the mixture in a microwave until agarose is completely dissolved. Note, the solution should be clear. Let it cool slightly for about 55 to 60 degrees Celsius to avoid warping the tray.
I'm Blessing Lawrence, and I'm talking about the step-by-step used to run a polyacrylamide gel electrophoresis. First, you need to know the materials needed, which is the acrylamide solution, the buffer, sodium sulfate, ammonium protein samples, loading dye, power supply, and staining solution. First thing is to prepare resolving gel solution and your stacking gel too. Next thing is prepare the samples, then you load and run the gel. After that is done, you visualize the protein and your sample is ready. Peace.
Oh, my name is Favor Dofia. I'm here to discuss on the step-by-step protocol on Agarose gel electrophoresis under visualizing the DNA. Firstly, if using a post stain, carefully remove the gel from the tray and place it in a staining solution, example ethidium bromide or SVBR safe, for 15 to 30 minutes and disdain if necessary. 2. If the dye was premixed in the gel or buffer, we visualize directly. 3. Place the gel on a UV trans illuminator. The DNA burns with fluorescence under UV light, that's the ultraviolet light. Also remember to wear a protective UV gloves or goggles. Thank you.
I'm David Goodness, and I'm going to be talking about the polyacrylamide gel electrophoresis, also known as PAGE. PAGE is mainly used for separating proteins and sometimes nucleic acids. There are some materials needed to run the polyacrylamide gel electrophoresis, and they are: we have the electrophoresis chamber, we have the power supply, we have our glass plates, we have the casting stand, and we also have the comb. Then we also have the staining solution and the staining solution. We have our protein samples and protein ladders. We also need the gel components like the acrylamide or bisacrylamide solution, the SDS, the TEMED, the APS. Thank
My name is Abigail Godsulobot, and I'll be talking on the step-by-step protocol on SDS, polyacrylamide gel electrophoresis. The first step is preparing the gel, which is into two layers. The stacking gel, which is at the top, 4% acrylamide, and the resolving gel, which is at the bottom, 8 to 16% acrylamide. Now, the stacking gel, large pore gels that stacks all protein into a sharp line before they enter the resolving gel, while the resolving gel is a small pore that actually separates the proteins by size. Now, the step by step involved in the stacking gel and the resolving gel. First off, the stacking gel. You pour the stacking gel, you pour off the overlaying solvent, you prepare the stacking gel mix. You add Temed, you pipette it on the top of the resolving gel and immediately insert the comb. You wait for it to polymerize. While the resolving gel, you mix the components, the water, acrylamide solution, trace buffer, SDS, which is the sodium diadesyl sulfate, and Temed. The Temed is added last to start the polymerization reaction. Quickly pipette the mixture between the glass plates, leaving space for the stacking gel. Carefully layer a solvent, example isopropanol or water, on top to create a flat surface. You wait for 20 to 30 minutes for it to polymerize.
My name is Blessing Nelson Achibo. Today I'll be demonstrating the basic steps involved in running an SDS page gel to separate proteins. SDS SDS page means sodium potassium sulfate polyacrylamide gel electrophoresis. I'll be talking on two steps, step 2 and 3. Step 2, prepare the samples. Mix your protein samples with SDS page loading dye. Heat samples at 95 to 100 degrees Celsius for 5 minutes. These denature the proteins and coat them uniformly with negative charges from SDS. Step 3, load and run the gel. Assemble the gel in the vertical electrophoresis chamber. Fill the upper and the lower chambers with running buffer. Carefully remove the comb.
Hello, my name is Good News Emmanuel Bassi. Here is a quick guide on how to visualize protein after PAGE, polyacrylamide gel electrophoresis. Number one is disassemble the gel plate. How do you do it? Carefully separate the gel plate to remove the gel. And number two is staining. You soak the gel in massive brilliant blue for 30 to 60 minutes with gentle shaking. Number three is the staining. Transfer the gel into a solution of methanol, acetic acid, and water to remove excess stain and reveal clay protein bands. Results: visible bands appear where proteins are present. Thank you.
I'm Eunice Isaac Kemedon, and I'm here to summarize the Agarose gel electrophoresis and polyclimary gel electrophoresis. The main use is separating the nucleic acid, which are the DNA and RNA, and separating proteins by molecular weight. Number two, the cross-linked polymer. The pore size like small, tunable, 100% acrylamide, and it is good for large size ranges. Very high, can distinguish 1 to 2D. It can distinguish 1 to 2 kDa differences. Now the typical setup: horizontal, the summer guide vertical between glasses plates, sample prep mixed with loading
My name is Inafo Nomana. I want to talk on the crucial safety net for PESH. First, we have the polyacrylamide. The unpolarized acrylamide monomer is a potent neurotoxin. Always wear gloves and handle with care. Second, we have the etidine bromide, a new mutagen. Wear gloves and dispose of waste properly. Third, we have the UV light. The UV light can damage the eye and skin. Always use a UV shield or wear protective goggle when visualizing gel. Fourth, we have the electrical hazard. Never open the lid or touch the buffer while the power is on. By following this protocol and understanding the principle, you will be able to effectively separate and analyze biomolecules central to biochemistry. Thank you.